So all of these various ideas can be brought together. We can consider how with multicellularity there comes concerns about how in fact you maintain cooperation among these individually replicating members of colonies. Now, for the most part, we're talking about cells when we consider microorganisms, but actually with larger organisms such as the social insects, bees, for example, ants, you've got a colony consisting of individual animals that are all cooperating with one another. And a lot of the same issues are seen there as well. How can you have these cooperating groups of individuals when it looks like the unit of selection instead should be, for example, the individual cell or the individual animal that is a member of these larger groups? So the answers to this is that cooperation tends to be achievable if you've got a number of different characteristics. So one of these are genetic bottlenecks. In other words, if the members of a group are closely related to one another, then you can have kin selection, you can have inclusive fitness. At an extreme, the members of a group can be clonally related to one another, and this can be achieved through a bottlenecking step that involves reducing the population size locally to just a single individual, and then creating the group of interacting individuals from the progeny of that single individual. If indeed you have an organism that reproduces asexually, then those progeny will all be clonally related to one another, and unless there are mutations that give rise to defection phenotypes, that is cheating, then you will have a group of individuals that at least potentially can all be cooperators. Or alternatively, and important, they might all be cheaters, in which case you can have competition going on between two different groups, and the cooperators can potentially outcompete the cheaters, where the cooperators are a group of individuals that are physically separated from another group of individuals that represent the cheaters. It's important to have clonality among the cells that make up microcolonies or other groups of cells, or again, if you have a group of animals that are interacting cooperatively, that at least they be closely related, which in fact is something you see among the social insects. It also is helpful if you have some kind of policing going on, and this is a means by which cheaters are in fact identified and removed from, or otherwise prevented from cheating, from within a group of cooperating individuals. And policing can involve also not just the prevention of cheating in closely related individuals, but also can be the prevention of cheating in individuals that are not closely related. And in fact, an important example of policing seen in multi-celled organisms is what we call the immune system, where individuals, whether they're generated by mutation, cancer cells, or instead are individuals that are parasites that come from the outside of these organisms, if you have a means by which these potential cheaters can be identified and either neutralized or eliminated, then you have the potential of retaining a group of individuals that make up a cooperating entity, being predominantly, if not entirely, cooperators rather than defectors. Thus, there is also concern over avoidance of invasion by parasitic cells or parasitic organisms, the avoidance of which is part of maintaining individuality and cooperation within multicellular assemblages. And the same thing again goes for social insects, where there is strong tendencies to identify and then neutralize or eliminate other individuals that have invaded a nest, because these individuals may be potential cheaters. They could be parasites, they could be predators of the social insect nest, and it's just best if you identify something that's in your midst that you know is not part of your own colony, to get rid of that individual. It's also important to have effective organism boundaries in addition to immune systems, although you can consider organism boundaries as in fact representing a part of the innate immune system as a means of keeping these cheaters from without from gaining access to the group of individuals that are associated as cooperators, such as making up microcolonies in the Kreft model sense. or instead, for that matter, boundaries that prevent individuals from invading the nests of social insects. Thus, for example, you have ants which might have a relatively small number of entrances to their colonies, and those entrances represent essentially boundaries that can be relatively easily defended. Or alternatively, you can think of everything that's not the holes as representing a boundary, where the holes are the only breaches in the boundary, and again, you need to defend these breaches in order to maintain the relative absence of cheaters from your midst within a nest. And so immune system, as noted, are also a means of preventing individuals that have in fact breached those boundaries to a large extent from being able to defect within the midst of a group of cooperating cells. But in addition to all of these ideas, there must be some compelling reason for greater size or complexity. And we've been through this idea. In order to have the selection favor an individual that in fact is larger or more complex, there have to be benefits associated with that larger size or complexity. So there's really two sides to this issue of the evolution of greater size and complexity. On the one hand, there is the reasons why an individual may benefit from being larger or more complex. And on the other side, which is predominantly what's being discussed in this slide, are the issues of the costs that arise once an individual becomes larger or more complex, and how these various costs are addressed. And we've considered this in a previous slide. What I'm pointing out here is a paper that I published in 2012 that walks through these various ideas specifically from the perspective of microcolonies and biofilms, and the utility that can come about from individuals basically remaining in each other's association to form aggregations, to form microcolonies, or to form cellular arrangements such that they potentially have fitness benefits that they would otherwise lack if they were just lone cells swimming in an otherwise non-spatially structured environment.